Hey everyone, my name is Scott Merritt, and this is the presentation that I've created to help you to find the results from the paper that I wrote about influenza and the dangers of antigenic shift. So, for a brief introduction, the reason I wrote this paper was because of the fact that influenza is one of the leading causes of epidemics and pandemics across the world. I don't know how far back some of your memories may go, but when I was a young lad, I remember hearing about and being afraid of things like the swine flu and the bird flu that were rampaging across the world and ended up thankfully not being as scary as things that we've seen recently, like the COVID-19 pandemic. However, were very scary to my young self. It's also interesting to note that we not only have this potential for pandemics and endemics with the influenza virus, but we also get our yearly flu shots because of its seasonal nature. Unfortunately, these influenza vaccinations are not always accurate and can have an efficacy rate of anywhere from 0 to 70%, with most falling down right at the middle at 50%. There are multiple different strains and types of strains of influenza, and while influenza, like all viruses, undergoes a process known as antigenic drift, which is somewhat similar to genetic drift, but different in the fact that it's point mutations in the genetic material of the virus due to improper copying and lack of proofreading, influenza also undergoes something called antigenic shift. So, antigenic shift, what causes it? Well, what causes antigenic shift is when you have multiple strains of the same virus that infect the same host cell and then swap their genetic material by accident when they're leaving the cell. Some viruses are capable of this, though the number is very limited. Most aren't, and that's because of something called a segmented genome, which means that the viral genome is broken up into little pieces instead of being one continuous thing, and these small pieces usually encode for one protein each. So, what's so different or unique about influenza? Well, like I mentioned, it's got a segmented genome with, as you can see in this picture, 8 different segments of negative sense single-stranded RNA, meaning that its genome is complementary to and opposite to normal human mRNA. So it needs to be converted to an mRNA type form before it can be translated. So each of these segments of the influenza genome encode for a different protein, with 2 of them encoding for a protein called hemagglutinin and another for neuraminidase, which make up the spike of the influenza virus. So hemagglutinin, uh, its function within the virus is to bind to sialic acid on the surface of the host cell, while neuraminidase, as the virus is leaving the cell to go and infect other cells, will actually cleave the cell sialic acid receptors so that the hemagglutinin doesn't stay stuck to the outside of the membrane. So if you have multiple strains of the influenza virus in a singular host cell, they can accidentally swap genomic segments during their packaging and exit, creating a new strain. And these strains are named by their hemagglutinin and neuraminidase products. So, for example, if you have H1N1, this means that you have type 1 hemagglutinin and type 1 neuraminidase within the virus. So, there are 2 main types of influenza strains, those that infect birds and those that infect humans, though typically one strain will not infect the other's host. This is due to the selectivity and specificity of hemagglutinin and its sialic acid target. So, to talk a little about sialic acid, we know that it's a monosaccharide that's found on animal cell surfaces, and as a monosaccharide, it usually is bound to galactose, either in what's known as an alpha 2,6 glycosidic linkage in humans or an alpha 2,3 glycosidic linkage in birds. So, what that means is, as you can see here with sialic acid and the galactose, you've got your carbon 1 and then carbon 2, which shares the bond here from the sialic acid. And on this galactose, you've got carbon 1, 2, 3, 4, 5, 6, and they're both facing in a very specific conformation. So therefore, it's alpha, and then 2, 6 glycosidic linkage. And in birds, it is the alpha 1, 2, and 1, 2, 3 that are forming that glycosidic linkage. And so, because of this, influenza virus, and because of its hemagglutinin shape, it serves as a very, very specific spike protein that fits in such a way that it can only bind to one type of sialic acid based on this glycosidic linkage, which is what prevents human influenza from infecting birds and avian influenza from infecting humans. So there was a study that was done by a group of researchers to show this property of hemagglutinin using human parainfluenza virus 1 and human parainfluenza virus 3. First, they had them infect normal human red blood cells to establish a baseline for the level of binding to sialic acid, following which they did cells that had increased levels of the alpha 2,6 sialic acid linkage, and they found increased levels of binding. And then they did it with cells that instead had alpha 2,3 sialic acid linkages, and they found in most cases decreased binding for human parainfluenza virus 3, though with one scenario where it was baseline, and for human parainfluenza virus 1, only decreased binding, thus prove or showing evidence for the fact that it is very specific on its sialic acid target. So how do novel strains have both avian and human influenza properties? Well, avian influenza and human influenza don't cross-infect hosts. However, they do share one host, which just so happens to be pigs. And so what's special about pigs? Well, most often they have an alpha 2,6 glycosidic linkage on their sialic acid, just like humans, but sometimes with certain individuals within a population, or even some populations, they have an alpha 2,3 linkage. And so this allows the pig to be infected with both types of influenza strains, both human and avian. And so they can then perform antigenic shift. And this produces combinations that aren't usually seen by the human immune system or the avian immune system. And therefore, it is much more dangerous to these hosts because of the longer time for an immune response to be mounted. So, in conclusion, influenza can perform antigenic shift with other strains of itself very easily by accidentally taking the wrong portions of genome when it is packaging. This can cause an uprising of new strains, which makes it difficult for vaccine synthesis due to the need to predict what kind of strain will be coming, not only strains moving through human populations, but also those moving through bird populations. And even more importantly, if you have both a human strain and an avian strain moving into a swine population, trying to detect or predict what will come out of that. Um, and though there are two different types of influenza that don't cross-infect, like we said, pigs act as a breeding ground for this antigenic shift between these two different types. And so, um, again, they need to be more aware of the strains moving through all these different populations when creating the yearly flu vaccine. And the best way to combat antigenic shift in influenza would be a universal flu vaccine to teach the body to fight against all of these different types of antigens and proteins that can come in the form of influenza. However, that comes with its own host of problems and difficulties and has been in development for years and will probably continue to take more years before we see anything in that regard. So this is my references cited page. Thank you so much for watching this. I hope I was able to help enlighten you a little bit more about influenza and the nature of antigenic shift. And I hope you enjoyed listening to this presentation. Thank you so much.